Hello, I am Margaret Pericak-Vance from the John P. Hussman Institute for Human Genomics at the University of Miami Miller School of Medicine. Today I'm going to present some recent advances in genetic studies in Alzheimer's disease in African Americans. I have nothing to disclose. Today I'm going to go over several new and exciting ongoing areas of research focused on genetic studies of Alzheimer's disease in African Americans. First, I will discuss some recent findings from Kunkel et al., which is in Press in JAMA Neurology. This study represents the largest genome-wide association study, GWAS, to date in African Americans. I will also briefly discuss some recent family-based genetic studies, as well as some gene expression studies. I will highlight ongoing studies using admixture analysis, but refer to you to the talk by Dr. Timothy Thornton in the APOE section of this conference for a more detailed discussion of admixture analysis. Finally, I will mention the ongoing efforts to increase diversity in our research cohorts by expanding the involvement of individuals of African ancestry in genetic studies. As we have heard, African Americans are more likely to develop Alzheimer's disease and dementia compared to non-Hispanic white populations. They are at greater familial risk for Alzheimer's disease, and they often have limited health care access, and thus AD patients are identified at later stages of the disease, resulting in poor treatment outcomes. If you have seen some of the previous presentations, you may recognize this figure from a 2019 study in South, which speaks volumes in terms of the lack of diversity to date in genetic association studies. It clearly indicates that the majority of studies have occurred in Europeans. This European bias has implications not only for risk predictions, but for future therapeutic developments. In fact, if we look at detail, we see that only about 2% of such studies took place with individuals of African ancestry. This lack of diversity extends to Alzheimer's disease genetic studies. The largest genetics genome-wide association study in African Americans, which I will discuss shortly, had only 8,000 individuals, whereas there were 93,000 individuals with European ancestry available for the 2019 publication by Kunkel et al. In Nature Genetics. So GWAS studies in African Americans have a power limitation because of numbers that compromise gene discovery, especially for rare variants associated with Alzheimer's disease. This figure shows that African American population is a two-way admixed population with Africans symbolized by the red and European, the blue, genetic ancestry. Admixture is a process when two or more isolated continental populations are interbreeding. On average, ancestry proportions in African Americans are approximately 80% of African, approximately 20% of European ancestry. However, as you can see in the graph, the percentage of European ancestry varies across individuals. As I noticed in the previous slide, there is considerable variability among African American individuals with respect to the level of European ancestry. Even more interesting is when we look at an individual and see how this ancestry is spread across their genome. Here in this slide, chromosome 1 to 22 are found along the y-axis, with genetic position along the chromosome on the x-axis. The colors represent the blocks of the genome inherited from their ancestors in this either European blue or African red. These 6 graphs represent actual participants in our research study, all of which have enrolled in the study as African American. They all have different admixture percentages. This concept of local ancestry around the gene is important in determining individual risks. This will be discussed in more detail in the APOE section of this conference. I'm not going to spend a lot of time on this slide, which I know you will see again, but to make the point that we know there are genetic distinctions between populations in Alzheimer's disease, thus emphasizing the need for more diversity in our genetic studies as we hunt for new treatment targets. Now I'd like to highlight very recent work in Alzheimer's disease genetics in African Americans. That is the paper entitled Novel Risk Loci and Pathways Associated with Alzheimer's Disease in African Americans, a genome-wide association study and meta-analysis. This paper is in press and will be published at the end of the month in JAMA Neurology. The study was led by Dr. Brian Kunkel from the University of Miami and Dr. Christiana Reitz et al. At Columbia University. This slide shows a comparison of the 2013 African American GWAS study led by Dr. Reitz for the Alzheimer's Disease Genetics Consortium, the ADGC, and the 2020 study. The 2020 study, as well as including nearly 3,000 additional participants, a 37% sample increase, also used the African Genome Research Panel for imputation, resulting in over 35 million SNPs for analysis versus 15 million in the previous study. While still underpowered to identify many rare variants, it did increase the resolution of the analysis. This slide shows two of the top novel results from the African American GWAS, including the top result, which is an intergenic locus near IGF-1R. The associated variants near IGF-1R, insulin growth factor receptor, are all African specific. Both expression and mouth studies support the involvement of this gene in AD. The second result I want to highlight is the RBFOX1 locus. This result replicates the association results from Reghaven et al., a recently published genome-wide association study of PET amyloid levels, which identified a novel locus for amyloidosis with RBFOX1. Variants in RBFOX1 were also associated with amyloid burden in autopsied brains, further supporting its role in AD. Pathway analysis of these GWAS data support the role of immune response, intracellular trafficking, and lipid metabolism. While the pathways are not ethnic specific, the variants included are: one novel inclusion from the African American data was the renal system development pathway. A multi-pronged approach is often necessary in order to deal with a complex problem such as Alzheimer's disease. To address the sample size and power limitations in case-controlled GWAS studies, we have also developed a family-based data set of multiplex, multi-generational African American Alzheimer's disease families, or the READY Consortium. We have identified over 80 multiplex Alzheimer's disease families of African ancestry. Since rare risk variants might be enriched in multiplex families, we performed family-based analysis on 44 of these families to identify rare risk variants. The strongest signal was found on chromosome 5 and was supported by a subset of 10 African American families. This region harbors two potential Alzheimer's disease genes, WWC1 and DOC2 genes. Both are involved in Alzheimer's disease pathology. Studies on the additional families are ongoing. These efforts confirm the value of family studies as an approach to decipher the underlying causes of Alzheimer's disease. Genetic variants associated with AD likely exert their effect by regulating gene expression. Previous population-based studies have shown that differences in genetic ancestry can have strong effects on gene expression. These differences have important consequences on the genomics of health and disease. Several recent studies have examined gene expression in individuals of African ancestry in Alzheimer's disease, including a study in gene expression in African Americans and non-Hispanic whites by Griswold et al., and one looking at RNA editing in the same populations by Gardner et al. The results of these studies show that differences in gene expression and RNA editing exist between Alzheimer's disease patients and controls in each ancestral group, African American and European. The specific genes are different, but the underlying molecular pathways converge. They are enriched for known AD pathways such as endocytosis and immune response. This suggests that an underlying common mechanism driven by distinct genes in diverse populations. These studies both underscore the importance of functional genomics works in diverse populations. With the increase in the number of African Americans involved in Alzheimer's disease genetics research, we are able to explore some additional approaches such as admixture mapping. We have analyzed 10,000 individuals from the ADGC and the Alzheimer's Disease Sequencing Project, or ADSP. And in our initial analysis using the admixture approach, we have identified a genome-wide significant result on chromosome 17. Interesting enough, these results confirm previously reported AD-associated region on chromosome 17, P13.2. This region includes the ZBTB4 gene identified by Blue et al. As an AD risk gene. You will hear more about the power and advantage of admixture mapping during Tim Thornton's talk in the ApoE section of this conference. I think these brief updates of this ongoing work stresses the importance of the inclusion of diverse populations in all research and highlights highlights the potential for gene discovery that will benefit all groups. I would like to briefly mention continued efforts in this area supported by the National Institute on Aging through the Alzheimer's Disease Sequencing Project Follow-Up Study, or the ADSP-FOOS, which is a global effort to generate whole genome sequencing on the path to drug discovery. Nearly 8,000 individuals of African ancestry are already included in the ADSP, along with individuals from a number of international cohorts from the Caribbean, South America, Asia, Australia, North America as well. New collaborative efforts include researchers in Africa, including Nigeria, Ghana, and Uganda, as we continue to increase individuals of African ancestry that will aid in gene discovery efforts in our path to develop new treatments and preventions. Thank you to my collaborators and to all members of the ADSP and the ADGC for their international efforts against Alzheimer's disease. Special thanks to the Alzheimer's Association and the National Institute on Aging for sponsoring this conference.